Hello everyone, my name is Malay Basu. I'm a faculty at University of Alabama at Birmingham, and today I'll be talking about our work on language of proteins. Ever since the Human Genome Project, the phrases like the book of life or the language of the genes have been used metaphorically. In this work, we took these phrases not metaphorically, but literally. In 2002, in this seminal paper, David Searles pointed out that linguistic metaphors have been woven into the fabric of molecular biology since its inception. We asked the question, do these analogies go deeper? And if so, can the methods developed for analyzing languages be applied to molecular biology? And this paper made me thinking that genomes can be thought of as book. The genes or the protein sequences can be thought of sentences, and protein domains can be thought of as words. So how similar are words and domains? In one of the figures in his paper, David Searles showed that the word distribution in Shakespeare's Romeo and Juliet is very similar to the domain frequency distribution in East genome. The plot that he was showing is called Zipf's law. It is a very important law in linguistics where the frequencies of the words are plotted on the y-axis and their sorted order are plotted on the x-axis. Zipf's law belongs to the power law family, where the plot becomes a straight line in log-log axis. If we draw Zipf's plot for domain frequency distribution in human genome, the slope is -1, which is identical to the value reported for English language. Before we proceed, we need to know about Engram Analysis. It's a very popular linguistic technique where consecutive words in sentences are considered together to find meaningful associations between them. The simplest Engram analysis is a bigram analysis where two words are taken together. We have already shown that a single word or domain frequency distribution in genomes follow power law. Not only single word distribution, but bigram frequency distribution in genomes also follow power law. And the slope of the curve is -1.8, which is very similar to the value reported for natural languages. So let me summarize that domains in genomes and words in natural languages are mathematically very similar. The first step in understanding any unknown language is Engram language modeling. Formally, Engram language models give the probability of a word given a set of preceding words. Depending on the number of the preceding words, called order, they can be unigram with 0 preceding word, bigram with 1 preceding word, or trigram with 2 preceding words, etc. The Engram models are also generative. Given Engram models from a genome, like human, we can generate a random human genome. To give an intuitive feeling, let's say that you are typing a word in Google search. The browser suggests alternatives based on the probabilistic association of the word, and that is based on Engram language model. The randomness of a system is measured as entropy. The relative entropy of a language model is the loss of entropy going from a completely random arrangement of words to meaningful word ordering on sentences, and this is dictated by grammar. There are around 7,000 languages in the world divided into 19 linguistic families. It turns out that all human languages have relative entropy of 3.6 bits. The reason behind this relative entropy in all human languages is not known, but it has been extensively used to find out whether a series of symbols represent a language, like the plaques discovered from the Indus Valley civilization. Formally, a bigram model is the conditional probability of a domain Dn given its preceding domain Dn-1. This can be calculated by dividing the count of the domain pairs by the count of the first domain Dn-1 in the genome. The problem with Engram language modeling is that the models are very sparse and it requires some kind of smoothing before they can be used. Regardless, we created language models from all branches of life, around 450 eukaryotes, 180 archaea, and 4,000 prokaryotes. So given the language model from this genome, the first question we asked is how much information is gained by the domain grammars of these genomes. Unigram entropy of these models is simply the sum of the weighted log values of frequencies of domains in the genome. This can be considered a randomized version of the genome as if all the domains in the genome are put in a bag of words and shaken. Bigram entropy is calculated similarly as the sum of the weighted conditional probabilities of the bigrams. And the relative entropy, or the information gained by the domain grammars in this genome, can be calculated by subtracting the bigram entropy from the unigram entropy. When we looked at the relative entropies in this genome, we found that except in metazoa or animals and a small group of archaea, the relative entropy lies in a close range around 1.2 bits. This is very similar to the universal relative entropy seen in case of natural languages. Now let's see how these grammars evolved. To investigate the evolution of these grammars, we calculated cross entropy or perplexity. It is a measure of uncertainty of a genome given the model from another genome. So the cross entropy of the genome G given the model M from another genome is calculated using counts of bigram from the target genome G, where the conditional probabilities come from the model M. The self entropy is calculated just like the cross entropy, but where the target and the source genome are the same. We can then normalize the cross entropy values by the self entropies and convert the values into distances. Evolutionarily distant species will have greater distances and similar genomes will have smaller distances. This results in a distance matrix which can be used to create a phylogenetic tree of the bigram models. We then created the tree for 38 eukaryotic species. The tree shows remarkable similarity to the existing evolutionary tree. Now, depending on the placement of the root, the tree conforms to either to crown group topology or the unicont and bicont tree topology. This shows that the bigram models have very strong evolutionary signals. We also use the bigram conditional probabilities to find the bigram features or phrases most prominent in each major branches of life. The bigram features identified important functions in each subgroups. Like eukaryotes and prokaryotes are differentiated by bigrams that are involved in ubiquitin and spliceosomes, the quintessential features of eukaryotes. We therefore conclude that like natural languages, protein grammars do have a constant universal relative entropy. There also exists a quasi-universal grammar of protein domains, and the 1.2 bit is the minimum complexity required to maintain a functional cell. The work described here has already been published in these 3 papers. Now let's move on to some unpublished work. We are now using this technique for large-scale modeling of protein families. Here we show that Engram model created from cyclin family from Panther database can correctly identify cyclins. We are now using this technique to identify antonyms and synonyms in proteome grammars. We are also investigating an unifying theory of origin of multicellularity using domain grammars. Using large-scale RNA-seq data as weights in the model, we are also investigating the complexity of tissue differentiation. Many people have contributed to this work. Particularly important is Eugene Koonin at NCBI. He taught me evolution. You might actually say my work has a distinct Koonian flavor. Lijia Wu from my lab did bulk of the work presented here. Now questions or attend to matters.